Hello, I'm Daniel Heath. I'm Emeritus Professor at the University of Windsor, and today I'm going to be talking about how omics technologies can be applied within fisheries management and fish conservation. So first, an overview of what I'll be talking about. I'll first define what omics are. I will then describe what sort of information or data can omics provide to a fisheries professional. And finally, I'll talk about how fisheries professionals can use omics to further their mandate. So what are omics? Well, it's a group of technologies that are based on the process of gene expression within the cell. And while you may have heard of genomics, perhaps not so much for transcriptomics, proteomics, metabolomics, methylomics, etc. And these group of technologies are collectively referred to as omics. So let's review just briefly how a cell goes about expressing its genes that leads to its ultimate function and form. And the function and form of the cell dictates what the whole organism looks like and how it copes with its environment. It all starts with the DNA, the genetic blueprint of the organism. And the information coded in that DNA is valuable for assessing what is the background of the organism. But you can go further in that in terms of expression of the genes, where individual genes are transcribed into short molecules called messenger RNA, and this holds the information necessary for actually expressing the gene. The messenger RNA is then translated into an amino acid chain, and in some cases the amino acid chain itself is functional, but more often the amino acid chain is folded and modified into a functional protein. And the protein is what contributes to the form and function of the cell and hence the organism. In some cases, the protein is actually further modified through metabolism into a metabolite. In some cases, the functional item is actually the metabolite. In other cases, it's a protein. And in some cases, the protein and the metabolite both have a function within the cell. So where does omics fit into this? Well, first of all, genomics is a study of the DNA sequence in an organism. And this provides information on similarities and differences in terms of the blueprint for individual fish or even populations of fish. If we go down this gene expression pathway, we next reach the messenger RNA or the transcriptome, where this is the pool of messenger RNA that reflects how the organism is expressing the genes coded in the DNA. So if you look at this, you might see more of messenger RNA for, let's say, immune function genes than you normally expect. And you could interpret that as maybe the fish was being challenged by a pathogen. The next is proteomics, and this relates to the pool of protein present in a tissue or a cell. And this provides information on the actual functional outcome of gene expression. And hence, it provides direct information about the form and function of the cell or the organism. Now, the transcriptome, messenger RNA, and the proteome, the protein, are very closely related, but they're not exactly the same thing because cells can abort the gene expression process between the messenger RNA and the protein if, for example, the cell does not require the final functional protein. And the last omics that I'm going to describe here is the metabolomics, which has to do with assessing the pool of metabolites that are present in a cell or a tissue. And this provides maybe more specialized information for specific needs when you're addressing a question related to fish health. Now, these steps within gene expression provide data through omics, but the question becomes, what is the level of accessibility of that information for a non-specialist fisheries manager or fish conservationists? Well, the early on applications, early on in the process of gene expression, such as genomics, are widely used within fisheries and conservation even now. So this is used for stock structure, for applied questions in terms of hybridization. And in fact, transcriptomics is also widely used within fisheries. So these are now applicable omics technologies. When you move further down the gene expression pathway to things such as proteomics and metabolomics, now you're talking about more advanced and complicated technologies, and although they certainly will have applications within fisheries science, it's perhaps not as prevalent right now. So because genomics and transcriptomics are the most widely used omics technology within fisheries and conservation, I'm going to give a couple of examples of genomics and transcriptomic applications. Genomics is primarily used, at least within fisheries and fish conservation, to assess stock structure or population structure. This information can be used to assess harvest levels. If you have a mixed stock fishery, you can use genomics to assign individual fish to one or other populations. When it's necessary to supplement a population of fish, you want to make sure that whatever your donor population is genetically compatible with your target population. And you can use genomic data to assess how similar or dissimilar these two populations are and choose the ideal source of supplementary fish. Genomics is also valuable for conservation status assessment. It can tell the user whether a population is inbred. It can tell whether the population is outbreeding with another population of fish, or even perhaps hybridizing with a different species of fish. And finally, the genomics can also be used to estimate effective population size of that target population. Transcriptomics, on the other hand, is primarily used to assess fish health. And this is done by assessing what is the pattern of gene expression. If there's elevated or reduced levels of expression of immune function genes, you might be able to assess that there is a problem occurring in terms of pathogen effects. Now you can assess fish health on an ongoing basis, and this is the basis of monitoring. And so in monitoring, you can either take samples from a wild population or a captive population, apply transcriptomic technology, and determine whether or not there are changes ongoing in terms of the pattern of gene expression. And this will give you information not only whether the fish are coping with the stressor, but also some indication of what is the nature of that stressor. In a more urgent application, you can use transcriptomics in a diagnostic function where you know there's something wrong with your population of fish, wild or captive, but you're not sure what. And the transcriptomics can lead you towards what is the possible stressor that is affecting the population that you're interested in. So clearly, fisheries conservation applications of omics are advanced. The technology is daunting, but in fact, the analysis and interpretation of those data are becoming easier and easier, particularly for genomics and transcriptomic part of omics. To help fisheries managers and fish conservationists navigate the potentials for omic applications, we've created a website that provides omics resources specifically for the non-technical oriented individual who's got a mandate for fishery management or fish conservation. On this website, we have instructional videos such as the one you're watching, infographics to define and describe omics, fish health decision tree. So this is what we've developed with the aid of omics experts for you to answer the question of, should I use transcriptomics to assess fish health in my application, or should I go with more traditional necropsy approaches? We also have a searchable published literature database, so if you want to go in and look at what's been published within the area of omics that you're interested in, that would be valuable. And finally, we have a list of omics experts who have agreed to be available either in a consultative or a collaborative role to help you with your application of omics. Finally, I'd like to ask that you take a few minutes of your time to complete a short Omics in Fisheries and Conservation survey. This will help us fine-tune the resources that we're developing for fisheries professionals in terms of applications of omics, and you can either use the web link or the QR code, and we would appreciate if you could do this survey. Thank you very much.